Okay, so welcome to this next video on the G-inhibitory pathway. Okay, so we're looking at pharmacological agents which interact with the G-inhibitory pathway, and at the moment we're looking at pharmacological agents which interact with the initial G-protein coupled receptors which start the whole pathway off, basically. So we've seen that three receptors which are all G-protein coupled receptors and are coupled to GI or GO heterotrimeric G proteins are the D2, D3 and D4 receptors. And we've looked at haloperidol, which is a competitive antagonist that's non-selective between the three, so it will bind to any one of these and it will block that binding site for dopamine and stop dopamine from being able to bind, and it itself will not stimulate catalytic activity in these receptors. Now, an example of an agonist of specifically the D2 receptors is cabergoline, and another example is metoclopramide. Okay, so metoclopramide, and I struggle with the name of this drug, metoclopramide. Metoclopramide is also a selective D2 agonist, i.e. it binds to the D2 receptor and stimulates the catalytic activity of that receptor, so it stimulates the G-inhibitory pathway, and so does cabergoline. So those are both selective D2 receptor agonists. Okay, so that's enough on the actual G-protein coupled receptors. Now what we'll do is we'll look at drugs which interfere further down. Okay, so two drugs that I want to mention are STHT31 and H89. So where do these drugs work? Well, basically STHT31, so this drug STHT31, what that drug is going to do is it stops the interaction between this second regulatory subunit of protein kinase A and the A kinase anchoring protein 12. So it's going to get in the way, if you like. It's going to bind to one of the two. I don't know which one it actually is. I suspect it's probably the A kinase anchoring protein 12. So it's going to bind and stop this interaction. It's going to, I think it binds to the A kinase anchoring protein 12, gravin, and then basically it binds over the site where the second regulatory subunit of protein kinase A would like to bind to the A kinase anchoring protein 12. And then what happens is that this whole R2C2 complex can no longer bind to the A kinase anchoring protein 12. And when that happens, this R2C2 complex dislocates effectively from the membrane. And if you've got no protein kinase A near the membrane, if you haven't got this R2C2 complex near the membrane, then you can't activate the protein kinase A by raising cyclic AMP levels close to the membrane. And this is a really important concept, that when you activate cyclic AMP production in the cytoplasm, you do not elevate cyclic AMP all over the cell. You elevate it in specific places. So if you no longer have protein kinase A in the correct place, that's going to seriously jeopardize cyclic AMP interactions with that protein kinase A, because cyclic AMP signaling is local. It's not a whole cytoplasmic event, it's a local thing, and if there's no protein kinase A near the cyclic AMP signal, then you're not going to get any further reaction. So STHT31 would basically produce a similar result to having the G-inhibitory pathway on continuously, basically, and reducing the activity of these protein kinase A molecules. Okay, now another drug is H89, and H89 basically inhibits the catalytic subunit. So it's a drug which binds to the catalytic subunit and stops it from doing its job. So even when the regulatory subunits of protein kinase A bind cyclic AMP, these catalytic subunits can go off, they can dislocate, but H89 will bind to them and inhibit them, basically, and stop them from adding phosphate groups onto serine and threonine residues of proteins. Okay, and the final drug I'd like to talk about is forskolin. And forskolin is basically a non-selective activator of adenylyl cyclases, so forskolin. and it activates all adenylyl cyclases except adenylyl cyclase 8. So, sorry, not adenylyl cyclase 8, adenylyl cyclase 9. So it activates all adenylyl cyclases except adenylyl cyclase 9. So basically what it will do is the exact opposite of the G-inhibitory pathway. It will elevate cyclic AMP levels and therefore raise protein kinase A activation. So forskolin is a very important drug, at least experimentally. It's not very important therapeutically.